Imagine, if you will, the possibility of getting on a long haul flight when we're able to fly again and using something that's like a nasal spray, but it would protect us against COVID-19. Or imagine being a frontline healthcare worker and being able to have just that sort of spray that would kill the coronavirus. Now, is this the stuff of science fiction or is it real science? I reached out to Aashish Manglik, who's a doctor and a scientist at the University of California, San Francisco, who's doing some fascinating research into this very area and has a breakthrough scientific discovery. Aashish, thank you so much for joining us.
Thanks for having me on, Sarah.
So you're calling this AeroNabs. Tell me, what is AeroNabs?
Yeah, so AeroNabs stands for aerosolized nanobody. And the idea here is that, again, there's a very small, very stable protein that binds to the spike protein on the surface of coronavirus. So the coronavirus is named, that corona comes from literally the outside shell of the virus, which is this protein called spike that latches on to the surface of our human cells by binding another protein called the ACE2 receptor. So that binary interaction, here's spike, here's ACE2, that binary interaction is absolutely required for the virus to get into our cells. So if one can block that interaction, that spike ACE2 interaction, then the virus basically can't get in.
It sounds incredibly promising. Does it work?
So we know that it's very effective at neutralizing the virus in a test tube. What we haven't figured out yet is whether it's going to be effective in humans. And that's what we're gearing up to do now, which is to do the kinds of experiments to learn whether it's safe enough to try in humans in the first place, and then eventually to see if it's effective.
And let's just be clear, when we do an investigation like this, or when you do an investigation like this to find these nanobodies, it's kind of the needle in a haystack, isn't it? How many did you look at to find what you were seeking?
What we do in my lab is we basically make a very, very large haystack. And our haystack right now is 9 billion individual nanobodies that we can then go look through. But once you find that needle in a haystack, you kind of have to sharpen that needle. You have to make it super sharp. And that's what we ended up doing in a very prospective chemical manner. So what we could get, for example, structures, three-dimensional atomic structures of how one of our nanobodies binds to the spike protein. By analyzing that, we could come up with very specific ways of making one of that nanobody better. So again, the best analogy I can come up with again here is, sharpening that needle and making it even sharper. And that's what we did. And that's what led to this incredibly tight and very stable molecule.
So when we're talking about that molecule, if we think of the human body as a house and coronavirus as a housebreaker, basically you're taking away all the housebreaker's tools. Is that right? You're basically deactivating all the stuff that makes the coronavirus able to get into you.
Absolutely, that's a great analogy.
So going forward with that, if this works, if you can get it from the lab and out into the world, what's your vision for how it could be used? How could it help frontline workers? How could it help travelers? How could it help people who are more vulnerable to this virus? We know that that's the case, that people of color in particular are more vulnerable to the virus. How do we make sure that something like this helps?
So very early on, we think that the biggest opportunity is for patients who've just learned that they have the coronavirus, but are not sick enough to go to the hospital. And right now, at least in the United States, what we do is tell those people to go home and self-quarantine. Some fraction of those people are going to get very sick, especially if they have predisposing medical conditions. So for those patients, we think that an approach like this, self-administering some way of, neutralizing the virus may actually prevent them from getting very sick. So that we think is probably the lowest hanging fruit for right now for what does an approach like this could offer. Now, the dream for us and for this entire research program is to really provide what we call pre-exposure prophylaxis, borrowing some terms from the work that people have been doing in the HIV space, which is for very healthy patients. So it could be everything from people who might get exposed to coronavirus, healthcare workers, maybe working on a coronavirus ward, or a traveler, like you mentioned, getting on a long haul flight that's concerned that they might get exposed to the virus.
How does this mesh with the vaccine? Is it basically complementary, or is it meant to take the place of the vaccine? How do you see it if it works?
We think of it as two ways. of course, what we all want and what we all need worldwide is a robust, widely deployable, and durable vaccine. And that historically has not happened in 12 to 18 months. If we pull that off, in some ways, it's one of the greatest achievements of biomedicine, at least in the past 100 years. But that's what we all want. So what we view this AeroNab approach as is, in the very short term, a bridge to that ultimate successful vaccine. And depending on when one of these AeroNabs approaches can reach patients, if there's still not a deployable vaccine at that point, then one of these approaches, these AeroNabs, would serve as a bridge to until a good vaccine comes about. But even in the context of a good vaccine, there might be segments of the population that are not good candidates, or where a vaccine may not be 100% effective. And that's where we think an approach like this would still remain quite complementary for the foreseeable future.
So if we go back to kind of the beginning of this research, it sounds like we have a lot to thank the llama for. What is it that the llama does that you have studied in your lab and so many others have studied? What's the link between llamas and nanobodies?
Yeah, so the deal with llamas and alpacas and camels too is that mostly, I think from an evolutionary accident, they make these very interesting pared down simplistic antibodies called nanobodies. And that's really, this was kind of a serendipitous discovery now almost 30 years ago by a group in Belgium. And over the past 30 years, the broader research community started to recognize that these simple little nanobodies are incredibly powerful proteins for a whole host of purposes. And so, as a result, my lab uses them for a whole variety of purposes, but they're really catching on broadly in the biomedical community.
All right, I have one more random question for you. If you go back to smallpox and the smallpox vaccine, my rusty memory of science is that people who worked with cattle didn't get it, or didn't get it as badly because they had been exposed to cattle. If I've got that right, is there any link to owning an alpaca or a llama and reducing your chances of getting coronavirus?
Okay, so this is actually a fascinating question that I've been digging into a little bit.
No kidding.
It turns out for MERS, yeah, so I don't know, there's probably not a great direct connection as in variolation, which has a beautiful history, and but there is, if you actually look at some of the cases of MERS coronavirus, which is the Middle Eastern Respiratory Syndrome coronavirus, yes. I think there is a hypothesis in the literature that one of the reservoir animals for MERS was a Bactrian camel. I'm suggesting that there may be something unique about how camelids control coronavirus, or at least in that case, maybe that camelid controlled the MERS virus, whereas other mammals perhaps have a more challenging time. Again, this is pure rampant speculation, riffing off of your very astute observation about variolation, but maybe there's a little something more to it.
Well, the bottom line is it's fun to dig into science with the experts. You are a doctor, you are a biophysicist, you are a chemist, you're an expert on this. If you were to make an educated guess with that kind of background, when we might get a chance to see your AeroNabs is something that we can put in our hands. Do you have any idea about when that might be, if everything goes the way you hope it will?
Realistically, if all of the stars align, I think it would be early 2021, something like that. But that's, again, with all the stars aligning and things really moving at the rate that one could hope for. The realism of really trying to do it in the current situation, every day is a different day and the landscape's changing, and trying to grapple with that is a different ballgame.
Is it exciting to be part of this massive worldwide hunt to make sure that we find answers and ways to protect ourselves against the coronavirus?
Oh, absolutely. There's in my relatively limited, but almost 1.5 decades of being in science, I've never seen the entire scientific community align on a singular mission, not just in one country, but, across the world. And it's really exciting to see this pace at which we're making new discoveries and the pace at which people can start building off of each other's work in real time. And just the frank openness of everyone to share their data to basically all get out of this mess as fast as possible.
For everybody who does long haul flights, not to mention all of those incredible people who are frontline workers, it sounds like exciting research indeed.